Good afternoon, dear students. Today we are going to start with a new topic, that is downstream processing, otherwise called as product recovery. What is downstream processing? The various steps or the various processes that are used for actually recovery of the useful product from the fermented broth is called as downstream processing. Okay. So the processes which are involved in recovery of the useful product from fermented broth is called as downstream processing, or in short DSP. Okay. Now the cost of downstream processing is often, or it is most of the time, more than 50% of the total cost of the production. Okay. So cost of DSP, downstream processing, it is a very critical process. Separating the desired product from the fermented broth, from the liquid, is a very critical step, and most of the time involves more than 50% production cost. Further, there is product loss and at each and every step of downstream processing. Okay. Now, most of the, and moreover, sometimes the product is present inside the cells or in the medium, or it may be present in both, in the cell or in the medium, and hence its concentration is also less. See, one thing, that there is product loss at each step of DSP. So the number of steps should be minimum to avoid product loss, right? Moreover, product is present inside the cell or in the medium or in both, and hence the concentration of product in the fermented broth is less. So, and the product needs to be separated from other molecules with which it is mixed. That means its purification is also needed. Therefore, downstream processing should be designed in a very efficient manner. Okay, so that there should be minimum number of steps to avoid product loss at each step, and it should be cost effective also. Otherwise, the final cost of the product may increase. Right. So therefore, downstream processing should be efficient, cost effective, and should involve minimum steps to avoid product loss. Now, what are the different steps involved in downstream processing? The first step is separation of solids and liquids. We also call it separation of particles or cell harvesting. Right. In our case, the solid part is the microbial cells that are present inside the fermented broth. So cell harvesting or solid liquid separation of particles. The second step is disintegration of cells or cell lysis, or breaking the cells. If the product is present inside the microbial cells, then we have to lyse the cells, break the cells to take out the product. So that is called as disintegration or lysis of cells. That is the second step. The third step is extraction of the product. The fourth step is concentration. If it is present in dilute form, we have to concentrate it. So after extraction, concentration of the product, then purification of the product, and then final finishing or polishing or drying, whatever is needed. So drying or finishing steps like polishing, etc. So what are the various steps of downstream processing? Solid liquid separation or cell harvesting, disintegration or lysis of cells, extraction of the product, concentrating the product, purification of the final product, and then polishing the finished product, or you can say finishing steps like drying, etc. Okay, so these are the various steps involved in downstream processing. Now let us one by one discuss various steps involved. The first step is separation of particles, that is solid liquid separation or cell harvesting. So removal of suspended cells from the spent medium or from the fermented medium. Spent means waste. Now the fermented medium is waste. So removal of suspended cells from the fermented broth is called as cell harvesting. So this is the first step in product recovery. What is the first step? Separation of whole cells, microbial cells or cell biomass, and other insoluble ingredients from the culture broth. There are several methods that are used for solid liquid separation. What are the different methods that are used? Flotation, flocculation, filtration, and centrifugation. What is it? Flotation, flocculation, filtration and centrifugation. What is flotation? While performing flotation, gas under pressure is bubbled inside the liquid broth. Okay, gas is bubbled into the fermented broth. Now, when gas is introduced into the liquid broth, it forms bubbles. So, bubbles are formed. The cells and other solid particles get adsorbed on the surface of these gas bubbles. Okay, when the because the gas bubbles are lighter in weight, okay, they rise to the surface of the fermented broth to form a foam layer. Okay, so when gas is introduced into the liquid broth, it forms bubbles. The microbial cells and other solids adsorb on the surface of the bubbles. The bubble rise to the surface of the liquid, forming a layer of foam. The foam layer can be easily removed. Right, now in order to form foam, we can facilitate foam formation. How? By adding certain substances like called as collectors, like long chain fatty acids or amines. Okay, so certain substances can be added to the medium so that foam is formed and stable foam is formed at the top layer. The foam contains bubbles. On the surface of bubbles, microbial cells are present. So foam contains cells and the foam is harvested, the foam is removed and cells can be harvested. So this is how flotation can help us in recovery of cells or harvesting the microbial cells. The second process is flocculation. Flocculation means forming floccules which are heavier and hence they will easily settle down. In flotation, cells are at the top. Cells are present on the foam bubbles. So they are separated from the surface. In flocculation, cells are separated from the bottom. So in flocculation, the cells or cell debris form large aggregates. They clump together. Okay, they form clump or aggregation. They aggregate, and large aggregates of the cell or cell biomass is heavy and hence it settles down easily at the bottom and it can be easily removed. So in flocculation, cells or cell debris forms aggregate. They settle down at the bottom and can be easily removed. The process of flocculation depends upon the nature of cells and the ionic constituents present in the medium. Okay, it depends upon the nature of cells and ionic constituents. Sometimes we add a flocculating agent like inorganic salts, organic polyelectrolytes or mineral hydrocolloids. So we add any flocculating agent which is, and after addition of flocculating agent, it is gently mixed. The liquid, the whole fermented broth is given a mix. And when the flocculating agent mixes, it forms large aggregate. Microorganisms collect on the along with the you can say flocculating agent like an inorganic salt, and the floccules, large floccules or large aggregates of microorganisms are formed with the help of flocculating agent and become heavy, and then they settle down, which can be easily removed from the bottom. So cells in case of flocculation are separated from the bottom. Now filtration. Filtration is the most commonly used method for separation of biomass or for separation of microbial cells and the culture filtrate. Okay, you all know what is a filtration process. You filter or you pass the solution, you can say fermented broth, through a filter, any filtration medium. The cells will remain at the top of the filter and the filtrate will can be collected from the bottom. So here we will filter such that microbial cells remain on the top of the filter and the liquid or spent liquid gets removed from the bottom. Okay, so filtration is the most commonly used method for separating of microbial cell, that is cell biomass and culture filtrate. The efficiency of filter depends upon various factors, that is what is the size of the microorganism, whether other organisms are present or not, presence of other organisms, then viscosity of the medium. If medium is liquid, it will be easily filtered. If it is viscous, it will not be easy to filter. Okay, so filtration efficiency depends upon size of microorganism, presence of other organism, viscosity of the medium, and of course temperature. Now several types of filters are used for filtration of the fermented broth, that is depth filters, absolute filters, rotary drum filter, which is again a commonly used filter, membrane filter, plate and frame filters, etc. Okay, now what are depth filters? Depth filters are made up of porous filamentous matrix. Okay, they are porous, they are made up of porous matrix like glass wool, asbestos, filter paper. Okay, so depth filters are composed of porous filamentous matrix like glass wool or asbestos or filter paper. When you pass the fermented broth through the depth filters, the particles are trapped within the matrix. The particles are trapped within the pores of the filamentous matrix, and the fluid passes out. Okay, so easy, composed of porous filamentous matrix. When you pass the fermented medium through it, the solid, that means the microbial cells or solid parts, gets trapped in the porous matrix and the fluid is separated out. So filamentous fungi can be easily removed by depth filters. Okay. What are absolute filters? Absolute filters are having specific pore sizes that are smaller than particles to be removed, of course. So absolute filters have specific pore size, a definite pore size, and of course smaller than that of particles to be removed. So when you pass the fermented broth, bacteria present in the or cells present in the culture medium will be removed and the filtrate will be, so the liquid and solid, that means cells and spent liquid, will be easily removed. So these filters have specific pore size smaller than particles to be removed, and hence when you pass the fermented broth through this filter, the cells will be easily separated. So bacteria that are present in culture medium can be easily removed by absolute filters because pore size is smaller than bacteria. Now the next is, or the more commonly used, rather you can say, is rotary drum vacuum filters. These are very efficiently used for separating filamentous fungi as well as yeast, larger cells rather we can say. These filters are frequently used for separation of broth which contains say approximately 10 to 40% of solids and the particle size is between 0.5 to 1 micrometer. Rotary drum filters are used successfully have been used successfully for the filtration of yeast, separation of yeast cells, filamentous fungi. Okay. Now, the filtration unit consists of a rotary drum. It is a circular rotary drum. Rotary drum means the drum which is rotating. It is partially immersed in a tank which contains the fermented broth. Okay, filtration unit consists of a rotating drum partially immersed in a tank containing the broth. As the drum rotates, it picks up the biomass. It picks up the microbial cells or particles, and the microbial biomass, microbial cells get deposited on the surface of the drum as a cake. As the drum rotates, it picks up the cells or particles from the fermented broth. It rotates in the broth. So it picks up the cells present in the broth and the cells get deposited on the surface of the drum in the form of a cake, and then the cake or the, you can say, deposited cells can be easily removed by scraping with knives. There are knives which will scrap the layer of cake which is formed on the rotating drum, which can be which is then dried and can be used. Okay, so the cells get deposited in the form of a cake on the surface of the drum, which are removed by scraping off with knife. The next is membrane filters. In this type of filtration, membranes with specific pore size can be used. So in membrane filters, we use filters, say cellulose acetate membrane filters can be used to separate the microbial cells, but they have a definite pore size. So depending upon their pore size, we can separate the particles. However, clogging of filter is a major limitation. The membrane filters having very fine pore size and they get blocked easily. There are two types of membrane filters that can be used, filtration: static filtration and cross flow filtration. In static filtration, the membrane is static. You just go on pouring the fermented broth and hence they get blocked easily. In cross flow filtration, culture broth is pumped in a crosswise fashion in the membrane. Okay, so you pump the culture broth in a crosswise fashion across the membrane, and this reduces the clogging process and hence it is better than static filtration. Now, what are the different types of filtration processes that are used? Again, let me tell you, filtration is the most commonly used method for cell harvesting or solid liquid separation. The three major type of filtrations based on particle size and other characters are used. They are microfiltration, ultrafiltration and reverse osmosis. These are the three types of filtration process depending upon particle size. Now, microfiltration is a process wherein the size of particles that are separated is 0.1 to 10 micrometer. Okay, and cell or cell fractions like viruses can be, okay, cell or cell fractions as well as viruses can be separated. Now the second, so microfiltration where is a process in which particles or cells of sizes 0.1 to 10 micrometer can be separated like cells, cell fractions or viruses. Ultrafiltration is a process in which we can separate particles of size 0.01 to 0.1 micrometer. Okay, again viruses, then cellular, subcellular components, subcellular fractions can be separated with ultrafiltration. Compounds with molecular weight greater than 1000 Daltons like enzymes and all, okay, can be separated. Now reverse osmosis, or also called as hyperfiltration, right, is it separates particles, size of particles that are separated by reverse osmosis is again smaller, 0.001 to 0.001, 0.0001 to 0.001 micrometers. Okay, compounds with molecular weight less than 1000 like sugars, particles and all, okay, these are all can be separated. Okay, so these are the three types of filtration. Depending upon the size of the particles, there's microfiltration, ultrafiltration and reverse osmosis. Now the last method of separating solids and liquids or separating the cells, harvesting the cell, is centrifugation. Centrifugation is a process in which the biomass is subjected to spin. It is spun, it is rotated in a centrifuge machine, it is centrifuged, and after centrifugation, because of centrifugal and gravitational force, the solids will settle down at the bottom of the centrifuge centrifugation tube and liquids can be separated. So centrifugation is a technique that is based on principle. Which principle? Density difference. Okay, separation by centrifugation depends upon the difference in density of the particles to be separated and the medium. Okay, so if the density of the particles or cell is more, they can be easily separated by a centrifugation process. So centrifugation is based on the principle that there is difference between the density of particles to be separated from the medium, and hence because there is difference in the density of the solid particles or cells and the medium, they can be easily separated by centrifugation. So centrifugation is mostly used for separating solid particles from liquid phase. Rather, you separate the solid liquid phase by subjecting the broth to centrifugation. Okay, now continuous in industries, continuous flow industrial centrifuges have been developed and they are continuously used. So you continuously feed the slurry, or you can say the fermented broth, and centrifuge it, and the solids will get separated and clarified liquid will be removed, and the solids that are separated get deposited at the bottom can be easily removed. Okay, what are the different types of centrifuges that are used? Okay, you can see the first one is tubular bowl centrifuge, then disc centrifuge, then multi-chamber centrifuge, and decanter or a scroll centrifuge. Okay, tubular bowl centrifuge, multi-chamber, disc, scroll centrifuge or decanters are used for the process of centrifugation. Now tubular bowl centrifuge. It's a simple centrifuge, small, commonly used in pilot plants. Okay, not you can say large scale commercial plants, but pilot plant fermenters. So on a large scale, of course, in industries. This is a small, simple centrifuge commonly used in pilot plants. Tubular bowl centrifuge can be operated at a very high centrifugal speed and can be run in both batch-wise manner or continuous manners. And the solids that settle down are separated manually. Okay. So tubular bowl centrifuge is a simple small centrifuge commonly used in pilot plants, can be operated at a high centrifugal speed and can be run in both batch or continuous mode, and solids that settle down are removed manually. The second type is a disc centrifuge, which consists of several discs that separate the bowl into settling zones. Okay, so the disc centrifuge consists of several discs, and the bowl, it separates the bowl into settling zones where the solids can settle. The slurry is fed into the bowl disc centrifuge through a central tube. The clarified fluid, that means the liquid part, moves upward while the solids settle down at the lower surface into the settling zones. Then there are multi-chamber centrifuges, basically a modification of tubular bowl type of centrifuge. It consists of several chambers, rather you can say several tubes, in such a way that the feed flows along the various tubes in a zigzag fashion. Now there is a variation in the centrifugal force in different chambers. This force is much higher in the periphery chambers. As a result, smallest particles settle down in the outermost chamber. Okay, so a multi-chamber centrifuge actually consists of many number of tubular ball centrifuges you can consider. So there are multiple chambers. The fluid is allowed to flow through the several chambers in a zigzag manner, and there is variation in centrifugal force. Highest centrifugal force in the outermost chambers, in the peripheral chambers, and hence smallest particles settle down in the outermost chambers and larger particles settle in the central chambers, in the chambers which are present in the middle part. Okay, so variation in the centrifugal force from one end to another end, leading to separation of even the smallest particles. Then the last one is scroll centrifuge or decanter, which is composed of rotating. The scroll centrifuge or scroll decanter consists of rotating horizontal bowls tapered at one end. Okay, so these are rotating horizontal bowls which are tapered at one end. The decanter is generally used to concentrate fluid with high solid concentration. When the biomass content is up to 80%, 5 to 80%, that is the solid materials are higher in concentration, we can use a decanter. The solids are deposited on the walls of the bowl, which can be scraped and removed from the narrow end. Okay, so you just pour the fermented broth within it. Because the solid content is very high, you have to spin it and the solids will stick up or get deposited on the walls of the bowl, and which can be scraped off after the liquid has been decanted or removed. So that is all with the first step of downstream processing, that is separating the solids from the liquids or harvesting the cells. Next we will discuss in the next class.